Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. Eric Jamois, Senior Director of Business Development, will introduce our speaker for today. In this pre-recorded webinar, Dr. Jeremy Jones, Principal Scientist, will discuss how artificial intelligence can be used in the drug discovery and development process to identify viable candidate molecules and shorten time to market. The webinar has been recorded in advance due to time zone considerations. However, Dr. Jones and Dr. Michael Lawless, Senior Principal Scientists, will both be available for the live Q&A after the presentation. A few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the question panel on your dashboard at any time, but we'll address them at the end, so after the presentation. All right, I'll pass things over to you, Eric.
All right. Thank you very much, Jasmine. It's my pleasure to introduce today's seminar speaker, Dr. Jeremy Jones. Jeremy has been at Simulations Plus for, well, about 6 months now already. Time flies. He was previously at several biotech companies in the US, and before that at the City of Hope Comprehensive Cancer Center. He has led multiple drug discovery projects with a strong emphasis on computational approaches, and he continues to do so at Simulations Plus, leading the AI-based, several AI-based drug discovery projects. And in this process, I'm also introducing our panelist, Dr. Michael Lawless. Michael's been at Simulations Plus for, well, over 12 years. He's currently a senior principal scientist, primarily involved in the construction and validation of our ADMET models, and that's really the foundation of our AI platform. So very happy to introduce both of them. We're going to start with Jeremy's presentation. Jeremy, the floor is yours.
My name is Jeremy Jones. Hi. Welcome to this webinar entitled Using AI-Driven Drug Design to Shorten Your Drug Development Process. My name is Jeremy Jones. I'm a principal scientist here at Simulations Plus. Quick bit of background about me. I got my PhD at Stanford in microbiology and immunology. I went and did my postdoc at UC San Francisco. I sort of transitioned into studying hormone nuclear receptors and oncology. Took that with me down to Southern California, where I was a professor for nearly a decade at City of Hope, which some of you may know is a National Cancer Institute designated National Cancer Center research hospital down there in the LA area. My lab studied drug development in urologic oncology. After my time there, I spent a short stint, about a year, in a startup biotech in the Pasadena area, doing sort of the same thing, drug discovery in the oncology space. And then in November of last year, I joined Simulations Plus, really to work on this AI-driven drug design platform. What I'm going to talk to you about today is a quick overview here, just how our AIDD module works. We'll go through sort of an example project where I'll give you a demo, working with a project near and dear to my heart, PPAR gamma inhibitors, something I've been working on for a while. And at the end, I'll just mention a couple other collaborative projects we have ongoing to design some other drugs as well. So, computer-aided drug design, in silico, de novo drug design, however you want to say it, has been around for decades. Though in the last, 5 to 10 years, with the advent of artificial intelligence and machine learning algorithms, we've really been able to increase our efficiency and productivity at designing drugs. So what does that look like at Simulations Plus? Some of you might be familiar with Simulations Plus. We're well known for our user-friendly software and providing expert consulting to support drug discovery, clinical development research, and regulatory submissions. I promise there's a reason that I'm mentioning all this. Our two flagship Software platforms are Gastro Plus, which is our mechanistically based simulation software package that simulates whatever route of administration, IV, oral, ocular, inhalation, simulates absorption, biopharmaceutics, pharmacokinetics, and pharmacodynamics in humans or animal models. And the other software platform is our ADMET Predictor, which is our flagship machine learning platform for ADMET modeling and property estimation. And it's also the home of our AI-driven drug design module. And again, the reason I mentioned this is because the AIDD module integrates the predictions from both ADMET Predictor and Gastro Plus into the generative chemistry process. And really, in my opinion, it's these sort of the best in industry predictions, property predictions that set us apart from other de novo drug design platforms. This module can be used in all the stages of early drug design and discovery, from hit discovery to hit the lead and lead optimization projects. And we'll show you some of that here. So what does automating the de novo drug design process look like with AIDD? Well, you're going to start with some seed molecules. And for instance, you can see here some chemical structures. And then the software will initialize with random and create randomly generated analogs using chemical transformations starting from these initial seed molecules. For each of those designed molecules, it's going to evaluate certain properties, and there's a large list of properties that you can pick from. Some of the typical ones we use are, we've created an ADMET Risk score, which takes into numerous different properties, predictions, into a single risk score. And I'm going to talk about in the next couple of slides some of these properties. We can predict the synthetic difficulty. Importantly, we can predict activity, or you can incorporate any user QSAR model if you want. For instance, I've built, and I'll show you how I built, QSAR models to predict PPAR gamma inhibition. And so you can use that to evaluate. You can do any number of high throughput PK predictions, for instance, predicting oral availability of compounds. And in the newest release, we have a 3D shape matching algorithm. So we can try to match up some of these randomly generated molecules to a 3D molecule of interest. After it evaluates these properties, the algorithm will prune the molecules using Pareto optimal layers. So picking the best of the molecules, and we'll talk a little bit about what that looks like. We'll then generate more analogs using those same chemical transforms, using randomly selected molecules from the current population as seeds for that next generation. We'll also take those Pareto optimal compounds and set them aside in the file. And the final, after you've repeated this how many ever times you want, a typical campaign would probably be 30 to 50 generations of this cycle. It will give you a list of these optimized molecules. So, as I said, I want to talk a little bit about some of these different properties and how AIDD works. The first thing I want to talk about is how we generate these analogs. We use a library of what we call chemically intelligent SMERCS transforms, SMERCS being a chemical encoding language. And if you wanted to find out a little more detail about this, you can see the link below. For instance, one of those transforms could be changing a non-fluorine atom to a fluorine atom, something that happens frequently in medicinal chemistry. The simple version of the SMARTS or SMERCS code might be something like something that's not a fluorine, changing it to a fluorine. However, you don't want that to happen just anywhere. You need to avoid things like changing the hydroxyl group in the carboxylic acid to a fluorine, so you don't get this highly reactive acid halide. And so we've worked long and hard, our medicinal chemists and cheminformatics folks, to make sure that these are intelligent transforms. And so you don't get things like that. And here you can see the much more complicated SMERCS code to make sure the fluorine only goes where you want it to. Currently, we have a library of about 200 transforms, but we're constantly adding on as things like new bioisosteres come out. We want to be able to incorporate those in any new information we get from our medicinal chemistry team. The next thing that happens is this multi-parameter optimization, right? And this is based on Pareto front calculations. Here is an example. For instance, if you had 1,000 normally distributed points, and if you look at this graph on the right, this is a 2D graph. So in this case, we're looking at two of those properties to be optimized. And this could be activity as a PIC50 and ADMET risk. And so what we'd want, or IC50 here, so we'd want a nice low IC50 and nice low ADMET risk. And what we'd want is to take those molecules that are optimized for these two parameters. And that Pareto front would be these 7 molecules. Now, if you're using 5 parameters, I have trouble envisioning this in my head, but in 5 dimensions, there would be about 100 Pareto optimal points. And it are those molecules that would be taken, put into that final file as optimal points, but also be the pool from which the seeds would be selected for the next generation of optimization. As I said, we typically optimize using 4 or 5 parameters. Some of the typical parameters are activity, a QSAR model. We can build in a docking score, really sort of anything you want. I'll show you that you want to build in, we can. The ADMET risk, synthetic difficulty, the high throughput PK, and the 3D shape matching are some other typical parameters. I did want to mention what goes into our ADMET risk, because I think it's a really nice way to measure a lot of important properties of a molecule in one score. And so here you can see things that go into the absorption, the size of the molecule, the number of rotatable bonds, hydrogen bond donors and acceptors. For distribution, we predict the volume of distribution, toxicity, whether or not it's going to inhibit hERG, whether or not it has any mutagenic potential. For metabolism, how it affects certain cytochrome P450s, and we predict intrinsic clearance as well. So putting all this into one score is really helpful for optimization. I also mentioned our high throughput physiologically based pharmacokinetic, or HT-PBPK predictions. And this is just a way of taking our top rated advanced compartmental absorption and transit ACAP model from GastroPlus to simulate dissolution, transit, and absorption in the GI tract. What's important here is that GastroPlus is really good at predicting these things throughout many different organs and throughout the whole body, but it does take minutes for each molecule, which is something we can't do. We want it to be much faster, so it can be incorporated into this AIDD, the generative chemistry process. And so what's great, in a real-world trial, we saw that the HT-PBPK approach produced comparable results to the full PBPK modeling using a range of molecules, but it reduced that simulation time from hours to seconds. Importantly here, this is purely in silico simulation. There's no experimental properties required to be input, and it does happen extremely rapidly. We do it in a multi-threaded fashion. And so here you can see 3 scatter plots that are comparing the HT-PK predictions to the full PK predictions. So you can see the area under the curve, the Cmax, and to a lesser extent, but still pretty good, the fractional oral available are pretty much dead on. And so it's really nice that we have confidence that we can predict these properties very quickly and add them as optimization parameters within AIDD. The last one I'll mention here is our brand new 3D shape matching algorithm. This was built by Dave Miller at Simulations Plus, just a fantastic scientist. And this is a way to try to match up the molecule in 3D space to a reference ligand. And so for me, for instance, in the PPAR project, there's a crystal structure with a known PPAR gamma inhibitor bound, and we can extract that 3D shape, the ligand in its pose, the way it is, and then use as a reference to try to see how closely in 3D space any of these AIDD generated molecules will match up. Not only that, not just in the 3D shape exactly, but you can also add into the similarity score a feature-based term as well. So here you can see some of the features in one of these molecules. You can look for hydrogen bond acceptors or donors, and positivity and negativity. So you, as the user, can balance how much weight you want to give to just the 3D shape or adding in some feature weights as well. And this has proved to be an invaluable tool in designing some novel molecules, some PPAR gamma inhibitors. All right. Now it's on to our example demonstration project of PPAR gamma inhibitors. I promise I won't spend too much time on the biology, but I am a biologist at heart. So one slide on PPAR and why we're interested in designing inhibitors. PPAR stands for peroxisome proliferator activated receptor. There's 3 isoforms. There's alpha, gamma, and delta. They are members of the nuclear receptor class of transcription factors, and all nuclear receptors sort of act this way. They bind to an activating ligand. They form a heterodimer. In the case of PPAR, it forms a heterodimer with RXR alpha. It recruits co-activators, and then it binds to specific response elements in the target genes. PPAR gamma is best known for regulating fatty acid metabolism, adipocyte differentiation, insulin sensitivity, and inflammatory responses. And you can see these are the kind of genes that it turns on. PPAR gamma agonists, drugs that turn on PPAR, and the major class here are the glitazones, are been widely used to treat type 2 diabetes by increasing sensitivity. Now, most of these drugs have been pulled from the market due to adverse side effects, including cardiac problems and also an increased risk of bladder cancer. And so about 5 years ago, several groups came out with papers in big journals identifying PPAR gamma as an oncogene in advanced bladder cancers. So about 20 to 30% of muscle invasive, these advanced bladder cancers, have mutations in the PPAR gamma pathway, either amplification of PPAR, point mutations in PPAR, or point mutations in RXR alpha that cause constitutive activation of PPAR gamma. And so the idea was that PPAR gamma inhibitors might be useful in treating these advanced bladder cancers. And in fact, just this month, there was announcement that the first PPAR gamma inhibitor is going into clinical trials for bladder cancer. So, the goal here is to design high potency inhibitors with good ADMET PK properties to sort of accelerate that pathway to the clinic. And I'm hoping, as we get into this, you'll see how easy it is to use this software as a tool to guide the drug design process. Again, the goal is to design this high potency inhibitor. What we're going to look to optimize is the activity based on QSAR models, which I'll show you in a second, the ADMET risk, the percent bioavailability, synthetic difficulty, and also 3D shape matching here as we do AIDD. Sort of the process for any campaign like this, or the strategy, is first we're going to build QSAR models that can predict activity using ADMET Modeler, which is another module within ADMET Predictor. And here I really want to hammer home the point that these models and the predictions are only as good as the data that you put into them. And I think that's one of the things I really like about ADMET Predictor and GastroPlus, is that our scientists really take a lot of time to analyze the data, make sure the data is good to make the best models, most predictive models. And so, for instance, you want to ensure that all the data is coming from similar assays so you can compare those data and perhaps benchmark compounds. Standard inhibitors have similar values from one lab to the other, so you can integrate that data. We're going to build both classification, a yes-no model, as well as a regression model, one that can predict the IC50 exactly. And then, as we'll get into, you're not going to just do one AIDD run. You're going to have different strategies within AIDD. For instance, for this project, I was looking to optimize side chains around that indole sulfonamide core. The next strategy was to actually take the best side chains and hold them constant, and then do some scaffold hopping, see if I can alter that core to make it better or more novel. And then 3D shape matching as well. And what's going to happen at the end of these runs is that you're going to compile the best compounds from these various runs, and then select the compounds that you think have the best chance of being active, and then synthesize and test them. And some things you can do here, I'll show you some of this. We'll generate classes, we'll assess some additional drug-like properties. Importantly, we're going to determine novelty and commercial availability, and we can rank with a multi-criteria decision tool. What does our, specifically into this project, what does our PPAR gamma inhibitor data set look like? Well, we don't have lots and lots of points, but for the classification model, we chose a cutoff of 10 micromolar. If there's any activity below 10 micromolar, we consider the compound active. If not, it's inactive. I was able to find 210 data points, 127 of which came from my old lab. And it was a pretty good balance between positive and negative, 90 negative, 120 positive. We have some tools in ADMET and Modeler that help us deal with unbalanced populations, but this is a pretty nicely balanced for building a classification model. For the regression model, we had fewer points, 113 data points, 62 of which came from my lab, that had reliable IC50 data. So the biologist in me goes and looks at the papers and says, yes, okay, this is how they did the assay. It's comparable with how I did it. I can combine these data. And what you can see is showing you some of the structures, a snapshot from ADMET Predictor. The data set's dominated by 2 to 3 classes, though there are some other molecules in there. You can see this sort of what I'm calling the indole sulfonamide substructure here with just a representative molecule. And those came from my lab. You can see this was called the quinoline ether, and there's 20 of these molecules that came from the literature. Sorry, there was 37 in the indole sulfonamide class. And then the next biggest class, there was 8 molecules that had this sort of 3 rings connected by either a sulfonamide or, a thiourea or urea. We have been more interested in the indole sulfonamide. You can see sort of the distribution of the PIC50s here. In this case, the higher value is better. So the PIC50 is the negative log of the IC50. So a PIC50 of 6 is equivalent to 1 micromolar, and PIC50 of 7 is equivalent to 100 nanomolar. So the higher the better here. And you can see some of these molecules have really nice high PIC50s. Classification model. Using ADMET Modeler, I built many ensembles of artificial neural networks. There's various ways you can build these models. The neural networks tend to produce really, really nice models. Used random stratified sampling to divide the test set and the training set. And what happens is the modeler will build, literally hundreds of different models with different numbers of input descriptors and different numbers of neurons, and then also provide some statistics for you to assess how good the model is. And some of the typical things we look at are a high sensitivity and specificity of both the training and test set. And here you can see really nice high values for both sensitivity and specificity. And when looking at this plot, you can see there's very few false positives, very few false negatives. Most of these compounds were categorized correctly. So a very nice classification model that we also used to inform our regression model, which had fewer points. Again, built many of these artificial neural networks and used the random stratified sampling. Here's some of the statistics we look at to make sure these models are good. We want a high R squared or Q squared value, a low root mean squared error value. And you want to see a nice, pretty linear distribution of both the training and test set along this line with that really nice range of PIC50s between, 5 and 9 here. So a really nice range. This model's pretty nice in the one we used in our AIDD. So finally, time to design some molecules. Back to this slide, our goal is in general to derive these novel high potency inhibitors with good ADMET PK properties. We're going to optimize based on activity and these other characteristics you see here. As I said, we'll get into some of these different AIDD approaches. And the typical settings for our builds would be 50 generations, with the number of molecules per generation. And we pair down with the best filters. But that's all something that you have the ability to play with and figure out what works best. That first strategy, sort of the directed sidechain optimization. So we need both a seed molecule and then I'll show you how this works, a query molecule. So our seed molecule is just going to be sort of the core scaffold of the sulfonamide. Here you can see that the core sort of scaffold here. And then really this could be anything, but for this particular run, I used a molecule that just had, these aromatic rings out here as a starting point for the software to build. Now, you can also add a query that will force any molecules that are generated to abide by that query. So, for instance, in this case, I wanted to make sure we kept this indole core, and we had these 3 different side chains, but they could basically be anything. And so what's nice is we can say, hey, this has to be this aromatic ring, has to be aromatic ring here too, these molecules have to be here. And then out here, this has to be an aromatic carbon. You have to have the one linker here to an aromatic carbon, and this can be any carbon here. And then I'm going to fill in these optimization parameters. It makes sense. So I'm going to switch over. I'm going to exit out of this now and switch over to ADMET Predictor so we can run one of these so you can see how it actually works. Okay, I've got our ADMET Predictor screen, as you see here. The software is up. Incredibly easy to use, a nice graphical user interface. But I want to dive into this here. You can see that seed molecule we were just talking about. You can double click and bring up Designer. Very nice editing software here. But here, again, here's that indole sulfonamide core. We're going to just start with 3 aromatic rings out here as our seed. I close that out. So it's as easy as this. We're going to come up here to start the AI drug design process. Click on that button. It's going to bring up a couple screens. The first of which is what you're going to select which properties you'd like to optimize. I've already pre-populated this just for a little bit of time, but you can see there's a bunch of different properties out here. Here we're going to optimize synthetic difficulty, fraction bioavailability, ADMET risk, and this is the QSAR model I've built to predict PPAR gamma inhibition activity. You also need to tell it which direction you're trying to optimize. Synthetic difficulty, we want it to be easier to synthesize, we're going to minimize. Same with ADMET risk, whereas we want to maximize the fraction bioavailable, and the PIC50 we want to maximize as well. All of the settings we pretty much use default. If you have some advanced needs, there's a lot of different things you can optimize, but the defaults work really well. You can pick the species for the PK property predictions. We predict both all 3 human, rat, and mice. So I just have it for human now, but if you were interested in what the bioavailability might be in mice for your xenograph model, you might want to switch over to the mouse. Let's move on to the next one here. Now here's where you can add in that scaffold query. I have the smart string of, where'd that guy go? Of this molecule again, because I want it to look like this. Get back over here. And so all the molecules that AIDD generates and that output will meet this scaffold. You can also have an input file containing product filter criteria. If there's some drug-like filters, if you have particular needs for your optimization or design process, you can add these here. All these files, we have some standard ones that we provide. They're very easy to open and edit if you're interested in specific things. So the next screen would just be here where we're picking the number of optimization generations. As I said, we typically do a 30 to 50. You might want some more candidates per generation in the initial population size, but for sake of time here, we'll keep it pretty small. The intermediate file frequency, this just, for instance, if you have 50%, it's going to write all the molecules that are at generation 15, and then again at the end at 30. But you could do it even more frequently than that if you want. It's kind of cool because you get to see how the molecules evolve over time. This is the file that contains all those molecular transforms. Again, this is something we provide, but you can always open it up and edit these files if you have ones you want to include or don't want to include. You can see all of the various ones we have here. We're just going to go with the standard. And then you're just going to select a folder here for the results. I'm just going to use this test. And if it's not empty, it'll let you know. So, all right, let's give it a try. We're going to hit run. No, it's not empty. Would you like to continue? Yes, that's fine, just because. And this also says that our seed molecule doesn't exactly fit that scaffold, which is fine. The software will deconstruct and reconstruct our seed molecule till it meets that scaffold. So that's fine as well. So we'll hit yes. And you can see how quickly it's going. It's already on generation 6, 7. It's generating all of these predictions for all of these molecules that quickly. You can see our population size is already up to 40 molecules. You know, there's a lot of randomness in this. So running this exact same, with the exact same settings again, you're going to get some different molecules each time. Let's see, it should pop up here. There we go. Pops up in a new window. And here's the output. I always like to make these rows a little bit bigger just because I like to see the molecules. But you can certainly see how this still has that indole sulfonamide core. And now out here, we have a methylphenol group, one with a carboxylic acid, one with a hydroxyphenol out here, some different shapes out here. And you can easily sort these. So these are the objectives. We were looking at oral availability. So, you know, the best had 95%. That's great. The lowest was 9, predicted to be 9%, maybe not so great. You can see all the different, now some of these are predicted to be really active from PPAR gamma. Some have pretty high ADMET risk. That's not great. So we definitely want lower ADMET risk. You know, you get lots of information. So it predicts, I'm just going to scroll across here. We're not going to look at all of these, but as you can see, we predict many, many properties and descriptors and calculate many descriptors as well. And there's a lot of room to add on. So I'm going to go back out of this for a second. We're not going to save this. I'm going to come back into our webinar. Let me make this big again. And so I just wanted to show you, for instance, when I did this run, what it typically looks like. We saw a variety of new side chains. You saw that clearly in the results that we just got. What I would typically do here is then perform I'd take out some of those ones that I really liked, and I would use those as seeds to perform additional rounds of AIDD with the best molecules from that run. One other kind of cool analysis I like to do is use a principal component analysis to examine how we've moved into different chemical spaces. So this is, for instance, a PCA plot from 2 different runs. The AIDD generated novel compounds here are in blue and red from an initial run and a secondary run. See, they from one run to the next, you move a little bit different chemical space, but this is compared to the compounds we used to construct that QSAR model in the green here. So you can see the AIDD runs really got us into some novel chemical space. I wanted to show you another strategy. I'm not going to do the AIDD for this one, but for instance, a scaffold hopping. For this, again, we used a seed molecule, but this time this was the most potent indole sulfonamide from our data set. So you can see these 3 side chains are a little bit different. Both have an aromatic ring, but have substituents coming off. And then I used a really complicated query here. And so, but it's basically a scaffold that forces this to maintain these 3 side chains. So basically saying all of these molecules are staying the same as they're drawn. And then I broke apart the indole ring here, and I just said I want this atom, and this atom to be part of an aromatic ring. So the idea here is hopefully that the software will keep all of these side chains, but then build out some different aromatic rings that might introduce, who knows, some new activity or some new novelty. I kept those optimization parameters the same. And what I saw, again, were the results were, you know, variations of that aromatic ring core. So let's pull this up just a little bit so we can see it better. So you can see we'd have some slightly different aromatic rings now. You know, a fused ring here with 3 nitrogens, two 5 members fused together with some substituents. And again, looking at principal component analysis, we moved away from, we moved into a new chemical space here as well. Just like before, I performed some additional AIDD rounds with the best molecules from this run, and then set them, put them aside so that we're going to compile them into best molecules from all these different strategies. One strategy I definitely wanted to show you in AIDD is our new 3D shape matching. And here, as I said, we're going to try to match the 3D shape to a reference ligand. For instance, you have a crystal structure, you can pull that out of the crystal structure. This is one of those indole sulfonamides I was talking about. If you're familiar with the PDB, the protein database, for instance, you can go and actually look at the molecules in the crystal structure. You can download the structure in the model pose, so not any ideal pose, but actually how it is binding in that crystal structure. So this is coming from a PPAR gamma ligand binding domain, and this is that molecule in 3D, how it looks. You can see it sort of here in the publication and what connections it's making. You can use various different seeds, and again, we're going to have the same optimization parameters. So let's go back to ADMET Predictor here. For this one, we'll just use the exact same seed we did before. We'll keep all of these optimization parameters the same, but now I'm going to hit this compute objectives using external application. And so this is where, if you're interested in putting in any of your external parameters, for instance, if you have your own docking program you'd like to use and build in those scores, you can call out to it here. We have a call out right now for our 3D shape matching. And it's really simple. You open up this document, and basically all you're going to do is plug in the link to where you've downloaded the 3D shape of your reference ligand. And so I have mine built in here. Everything else is going to stay the same. You can add a scaffold query if you want. I kind of like not having the scaffold query here. It really allows the software to go in some different directions. In the 3D space, we're going to keep that same drug-like filter just to make sure we're not getting anything too crazy. Again, the input file with the transform rules, the folder for the results, we'll keep it the same. It'll tell me that it's full, but we'll overwrite it. We're going to go just a couple generations here because it does take a little bit longer with the 3D 3D shape matching optimization. So let's give this a try. Yes. And you can see it takes a little bit longer in each generation because for all those molecules it's creating, it's doing the 100 different conformations and trying to match them up. So we're on to generation 2. You can see it's a little bit slower, but honestly, none of these runs, even for 50 generations, is going to take more than 5 or 10 minutes. And if you had even a really computational intensive call out, you could set it to run overnight if you wanted to, and wake up the next morning with a whole list of your optimized compounds. So now we're in the final population, and it'll open up a new window here in just a second. And the reason I'm doing this is because I want you to see some of the cool things that pop up here in the new window. Here we go. And so again, you see some cool molecules come out. You can sort by the different objectives. The new objective here is the 3D similarity. And so closer to 1 would be the most similar. This one is about 74% similar to our reference molecule. And I'm thinking that reference molecule has a couple other things here. So this is kind of a simplified version of it. Let's take one that's a little bit different and see how it overlays. Let's try this one, 72%. So one really cool thing is if you have a 3D visualization software, you can just put the link to it in ADMET Predictor and our software will automatically generate a link to do the overlay. So here, the NSI, this is the reference ligand from the protein database. I'm just going to click on this to turn it gold. And this is the molecule from AIDD and how it overlays. You can see that it's keeping this indole sulfonamide, and the overlay there is really nice, or the indole. And the side chains, the aromatic rings, overlay pretty nicely here. And you can see, this is just a more simple aromatic ring without this substituent. This was just, a really first pass quick. It's still a pretty nice result. I will show you some of the results I got from when I spent a little bit more time working with this. We got, very good results in terms of similarity and activity and ADMET PK properties. Here I'm just showing you one of the ones I got from my run. It did some interesting things here with the 3D shape matching. This is the 2D ligand for comparison. And this is the one AIDG generated. You see it has about 86% 3D shape similarity, still predicted IC50 of about 100 nanomolar, so that's good. And you were able to change, for instance, this trifluoro, the CF3 group out here. Now instead you have this fused ring that takes up very similar space. So you get some really cool molecules. And as I showed you, you automatically generate that 3D overlay. In a way, this is almost can be used, thought of as an alternative method for scaffold hopping, because you really can get some different things that come out of here. And we did get some molecules that didn't have this indole here and didn't have a different scaffold. So on to sort of the last step here. How do we select compounds for study? Again, the overall goal was the high potency inhibitors. I wanted to add one here. We wanted it to be novel. And so we do some novelty searching too. But the process, we're going to combine all these compounds from all the AIDD runs that meet whatever criteria you want. For me, what was important to me was I wanted the PIC50 to be more than 6, so sub-micromolar, had an ADMET risk score of less than 6, be at least 40% bioavailable. I know from practice some of these molecules aren't so soluble, so I was selecting some with better solubility so I could work with them in my experiments. Synthetic difficulty score of less than 5.5. And so with all these runs, that led me to have 181 compounds meeting all these filters. I actually took these, I went and performed docking studies and had docking scores to help me figure out which ones I wanted to focus on. I was able to separate these into classes, and that really helps us to understand potential SAR and select some diverse candidates to test. I also searched for novelty and commercial availability. These searches are currently post hoc, but we're working hard to integrate these as optimization parameters so that we can ensure, without having to do a second step, that the compounds you're working with will be novel and patentable. And if you're interested in some quick commercial availability, there's a huge chemical space, synthesis on demand space out there of over 1 trillion compounds, places like enamine that you can order from and get them within 3 to 4 weeks. So we want to be able to tell you if there's any commercially available compounds that you can just order and don't have to work to synthesize. We also have a multi-criteria decision making tool to help you prioritize compounds, and you can weight these and put whatever parameters you want in. When you have compiled this list, for instance, I put a lot of weight on predicted PIC50 and the 3D shape similarity, and novelty, and to a lesser extent the docking score. We calculate additional drug-like measures that are common in medicinal chemistry, the lipid ligand efficiency, the lipid metabolic efficiency, the ligand efficiency, log P. So I used those to help me decide which ones I wanted to focus on. And then you order the available compounds or discuss the other candidates with your synthetic chemistry team. Order them, synthesize them, test them, and ideally you'd go back and maybe modify and rebuild and improve your QSAR models and proceed to the next stage of optimization. I will say that, again, this is new. We don't have any of these molecules yet tested, but success stories are on the way, including this PPAR gamma inhibitor one. As I said, it's near and dear to my heart. I'm really hoping to identify some novel potent inhibitors. But we do have some collaborations that are ongoing. This one is actually finished. We're just waiting to publish. We partnered with a large pharma company. They were kind enough to provide us with compound structures and activity data from one of their ongoing drug discovery programs. We worked with them to define the multi-objective parameters against which the molecules needed to be optimized, and use their existing SAR data to build our neural network user activity models. I'm just going to give results in general terms here, nothing specific yet. But the company synthesized and tested 70 compounds selected by their sort of traditional in-house medchem approach, their medchem team, and then 23 compounds that our team picked using our AIDD campaign. Again, no specifics here, but a higher fraction of our AIDD design compounds met the company's success criteria, which was a mixture of activity and ADMET properties compared to the in-house, so we were happy with that. We also generated follow-on libraries of virtual compounds with optimal predicted combinations of these properties for further screening and analysis for them. And as I said, we're hoping to publish these results in the near future. I'm almost done, but I did want to mention that we've also just started 2 new drug discovery partnerships. One with Sino-American Cancer Foundation to work for inhibitors of the MTHFD2 protein, and another with the Polish Academy of Sciences, a researcher there, to work on another nuclear receptor looking for ROR gamma ligands. So we're excited that these projects are underway, and we're excited to see the results and how well our predictions bear out. Just so you can see kind of what one of these projects looks like. You know, basically when we work with one of these collaborators, you sit down with their team. We sit down with their team. We kind of review the target, curate whatever relevant literature and data is out there to help build one of these QSAR models, or integrate them if they already have them in the case of that big pharma company. We'll do some initial medchem assessment. We'll, do our AIDD campaign, looking for novelty and availability. We'll do additional rounds of AIDD as necessary. And then we come back to the collaborator and say, hey, here's the ones we think look good. We can help them with facilitating purchasing, or set them up with medicinal chemists that we know to help them with their project. And then they'll test the compounds, and hopefully some of them look good and they still want to keep working with us. We'll go back and do additional rounds as necessary, or move on to the next stage of the project. So last slide here. Take-home messages. Artificial intelligence approaches can really accelerate the drug discovery and optimization process. Though I will say there are many of these out there now. I'd say the results are only as good as the data and the models on which they're built. Again, I think that ADMET Predictor has some of the best. The AIDD module that is built on these predictions within the ADMET Predictor platform is very user-friendly. It's great for end-to-end drug discovery and optimization. We're also here to help if you don't want to use the software directly, you want to partner with us to do some of this drug discovery. We really like doing that. I really like doing that. The final message I'll leave you with here is that drug discovery, despite all these awesome tools, is still an intensely human activity, and will remain so for the foreseeable future. We don't see these AI drug discovery platforms replacing experts, but instead we see them as a tool to help with decision making, to speed up the process, and just as an additional tool. What we hope to do is prompt a mix of responses when we give our partners a list of potential molecules. And medicinal chemists on the team might say, "Oh, of course, that makes sense." Well, maybe, and now that's interesting. Maybe something they hadn't thought of. Just as a great tool to work with the team to do drug development and discovery. So with that, I'll say, Tēnā koutou katoa. That's I'm actually live in New Zealand at this point, and that's how we would say thank you, everyone, in Maori. But specific thanks to members of my team, Michael Lawless, David Miller, Rafael, and the entire Chem Informatics team. So with that, I will end, and now I'll be available to take questions. Thank you.
All right. All right, great, Jeremy. Thank you so much for this very nice presentation, very good tour of the AIDD capabilities. Yeah, there's a very significant number of questions. I did my best to answer the more pinpointed ones directly through the chat, but there are others that are really of more general interest that that I thought, you know, I would submit to you and Michael. Some of them are actually questions that, you know, come back to us, you know, fairly often. So the first one is for a QSAR model, how many compounds are enough to make a good model? And does it require the same series of compounds? I think so. So I think maybe there's kind of two different things here. I mean, what we built as models are intended to be global models for the most part, and what maybe, so we use these as part of the IDD framework in many instances, but customers may also be interested in using local models that are more relevant specifically to that series of molecules. So the question is probably more, you know, specific to that. If you're going to build local models, how many molecules, sort of ballpark, you would need to build a quote unquote good model, which I would interpret to mean a model that steers the design in the right direction. I think that's the main idea in the IDD, is to steer the optimization of the structures, you know, towards compounds that, you know, fit these models. I mean, fit the directives, the guidelines better. So
yeah, if you don't mind, I'll chime in too. Like speaking specifically to like the activity model, the PIC50 models I built. You saw in my data that there was a couple different sort of series, and I will say that I built models for each individual series and then combined just to kind of compare how they performed. And honestly, in a couple projects now, combining different series that, again, important that they're built on the same type of assay so that it's fair to compare those data. Having that wider array of compounds actually made better models. If you're looking for a minimum number of compounds you need, you know, as always, the more data is the better. You're going to get a better model. But no, you saw, you know, I built really nice models with around 100 compounds or so.
Yeah, I think, you know, some people have probably built some with 30 or 50, but I mean, this is really kind of on the edge of what's, you know, then you need to have enough coverage to, you know, build something meaningful. And, you know, I think we'll also get many times the question about the, you know, the absolute values you get out of the model, as opposed to, again, being able to, the model being good enough to just steer the design in the right direction. And that's also that there could be a systematic error in the prediction, but that is not, from what I understand, that important, because as long as it's consistent toward the molecules, then you're still steering in the right direction in terms of improving your design towards the objectives. All right.
Yeah, I'll just add that there's no hard and fast number of compounds, but you also have to consider the range of activities. And typically, I say 3 orders of magnitude. So if you had say a 0.1 micromolar compound, you'd need to go up, you know, 3 orders of magnitude above that for the range to get a good QSAR model. So you also need to consider that with your data. And the other thing is you'll get more and more data as the project proceeds. So you're going to generate compounds to synthesize and test, and then once they're tested, you can add them into your QSAR model for future model.
Yeah, it is nice to do that sort of iterative approach to, you know, do a round, test them, and then build onto the model. There's a related question in the Q&A too, Michael, that asks, you know, can you build a QSAR model for compounds in the micromolar range, which don't share the same chemical space, but also be able to predict new compounds whose activities in the nanomolar range? And It's possible, but as Michael was saying, and Michael, I'll let you answer this too. You're generally building a model, and you're going to find compounds within the range of the data set from which you're building.
Yeah. Yeah.
Oh, go ahead, Eric.
No. I think that's very fair.
Yeah, the next question here, I was thinking about while I read it was for the whole AID workflow. Can we skip the QSAR model generation and start directly from something like a docking score? And the answer is absolutely. You don't need a QSAR model to run AIDD. Certainly, if you're pretty happy with what you know about the molecule already, you can optimize other things, including the docking score. So absolutely, you can start directly from a docking score. Mm-hmm. And I'll just say here, I apologize if my brain is a little bit slow. You saw that I pre-recorded that, and it's because, as I said, I'm in New Zealand where it's 5 in the morning here. And so I appreciate also Michael and Eric being on the call to help me with answers as I'm still waking up.
Mm-hmm. Yeah, so no, we appreciate that, Jeremy. Thank you. There's a pretty cool question here about the chemical transforms and to what degree you can create your own transforms. I think it's very pertinent to the flexibility of the platform. So maybe you can comment on that.
Yeah, absolutely. And the answer is yes, you can create your own transforms. For this, in fact, it's incredibly easy. You just open up our transform file and add a line you want. For instance, for this specific project, I actually added a specific transform that sort of tweaked a little bit how we could add a sulfonamide directly to nitrogens in certain ways. So yeah, very, very easy, user-friendly to do. Absolutely.
Thank you. Thank you. Also an interesting question on the reproducibility of the results concerning the use of random seeds. I know that is something if you want to get the exact same answer, you probably do want to use the same random seed in your run.
Yeah, there's one other thing you have to do too, for sure, which is take off the multi-threading. So the multi-threading allows it to run in parallel process and really speeds up the run, but doing so introduces some randomness. So if you want absolutely reproducible results, same random seed, take off the multi-threading. Right, Michael?
Exactly.
Yeah.
Perfect.
The question here, how do you make the QSAR models transparent?
I'm not sure how to read
Yeah,
that.
Michael, any thoughts on this one?
Well, there'll be a net file. I mean, obviously, if you want other people to use your model, you can publish it, but they'd have to, have ADMET Predictor to do it. That we have what's called a net file, and it has the descriptors and is the basis for the A and E model. But it'd be hard to kind of take that and recode it. So it'd be best just to give someone your net file, which is created part of the model that encodes the model, and then have them access that ADMET Predictor.
One thing I really like about the models we build here too is that you can go back and see what the descriptors were chosen, what our modeling software used as the descriptors. Not only that, you can go into ADMET Predictor and do something. We have a tool called structure sensitivity analysis. So for instance, with my compounds, there was one of the descriptors was I think it was anion charge, right? And you can go and see what parts of each molecule contribute negatively or positively to a specific model. It could be for ADMET risk, or it could be for solubility, or it could be for your QSAR model. And so it's a nice way of looking at each molecule and saying, oh, okay, this part of the molecule is negatively influencing my IC50. Whereas this other part of the molecule is contributing to it. So it's a really nice tool for understanding, really understanding the structure-activity relationships. I know that's a little bit of a digression, but it's one of the tools that we have that really makes so the medicinal chemistry process accessible to everybody. As you saw, that I'm really a biologist by training who really enjoys this stuff, but all of these tools make the software very, very accessible to everybody in related fields.
Cool. Well, thank you. I think we're at the top of the hour. I don't know. Going to ask Tanya if we can take more questions or if we need to wrap up at the top of the hour.
There's one real quick. We'll just answer in case of drug repurposing studies. What would be the workflow? Virtual screening, the molecular docking, and then AIDD. I would say this is a little bit tough because you want to start with known drugs, and so you would. Yes, this is a possible workflow. The problem with AIDD is you're going to get new molecules that aren't already accessible drugs, but you could use ADMET Predictor to help identify which of those repurposed drugs would have the best characteristics for your indication. For instance, if you needed the drug to get across the blood-brain barrier, we have a good model to predict whether the compound will get across or not.
Yeah, that's a good.
I'll just say thanks, Ben, for the shout out. Ben was an old member of my lab at City of Hope.
Nice. All right. So I just want to thank everyone. Thank you, Jeremy, Michael, Eric. And then again, for any questions we didn't get to, we will follow up with you. And then we invite you to learn more about AIDD or other products and services on our website. We also do offer a free one-year access to Simulations Plus software to students and educators at accredited universities worldwide. We invite you to follow us on social media. And then this webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you so much.
Thanks to everyone. Have a great rest of the day.
Bye-bye, everyone.
Bye-bye.